let's microbiology it. Our first micro mind map, it's all about physical growth factors. Stemming from that, our first branch is temperature. And then we have psychrophiles, mesophiles, thermophiles, and hyperthermophiles. Our second branch, it's pH. Stemming from that is acidophiles, neutrophiles, and alkalophiles. Our last and third branch is osmotic pressure. Stemming from that, it's hypertonic, hypotonic, isotonic, and halophiles. Microbial growth is tightly regulated by physical and environmental factors, primarily temperature, pH, and osmotic pressure. These factors directly affect enzyme structure, membrane integrity, and metabolic efficiency. Because microbial enzymes are highly sensitive to environmental changes, even small deviations from optimal conditions can drastically slow growth or cause cell death. Temperature classification reflects evolutionary adaptation. Psychrophiles thrive in cold environments by producing enzymes with high flexibility, while mesophiles, which include most human pathogens, grow best at moderate temperatures near 37 degrees Celsius. Thermophiles and hyperthermophiles, commonly found among archaea, possess heat-stable proteins with membranes that prevent denaturation at extreme temperatures. pH affects microbial growth by altering hydrogen ion concentration, which impacts protein folding and transport systems. Most bacteria prefer a near neutral pH, that's 6.5 to 7.5, while acidophiles thrive below pH 4. Fungi, such as molds and yeast, are adapted to slightly acidic environments, giving them a competitive advantage in food spoilage. Osmotic pressure determines water movement across the cell membrane. In hypertonic environments, water exits the cell, causing plasmolysis. In hypotonic environments, excessive water influx can lead to cell lysis if the cell wall is weakened. Specialized microbes such as halophiles thrive in high salt conditions by maintaining osmotic balance. Now that we have a little more information, let's fill in our micromind map, starting with psychrophiles. They can grow at 0 degrees Celsius and have an optimum temperature of 15 degrees Celsius. They can be found in the deep ocean, deep sea, or in polar regions. Next, we have mesophiles. Mesophiles have pathogenic bacteria that can grow in 37 degrees Celsius. Their range is 25 to 40 degrees Celsius, and they are associated with diseased organisms and food spoilage. Thermophiles. Their range is 50 to 60 degrees Celsius, and you can find them in hot tap water, hot springs, and sunlit soil. Hyperthermophiles. You can find them in hot springs and hydrothermal vents. Their temperature range is about 80 degrees Celsius or higher, and some archaea are hyperthermophiles. On to pH. Acidophiles. They have a pH of 4, and mold and yeast have a pH between 5 and 6. Neutrophiles. They have a pH of 6.5 to 7.5. Alkalophiles have a pH of 10, and their range is 8.5 to 11. You can find them in soda lakes, alkaline soil, and alkaline hot springs. Onto osmotic pressure. First one is hypertonic. So what happens on the outside of the cell? Well, there's an increase of solute. In the cell, water leaves. Also associated with hypertonic is plasmolysis. It is the breaking of the plasma membrane. Hypotonic. Well, inside the cell, there's an increase of solutes. Outside the cell, water flows inside the cell. Next, we have isotonic, which is the net movement of water. And then we have halophiles, where you can find them in the Dead Sea, and they can survive in salt concentrations 25% or higher.